Hi everybody, thank you again for joining Medical Devices Cellular Imaging webinar series. My name is Grischa Chandy and I'm the host for today's webinar. I'm the product manager at Medical Devices for the cellular imaging instrumentation and software. We also have joining us DeLaura from Cellular Dynamics International. Susan is a product manager at Cellular Dynamics International, where she manages the human iPSC-derived neuron and endothelial cell line. Susan's professional background also includes research in molecular and cell biology at the University of Wisconsin-Madison Comprehensive Cancer Center. Additionally, we have Jayne Hesley, and she is the application scientist in the cellular imaging group at Medical Devices. At any time during the webinar, please submit Q&A questions through the Q&A window. To do that, you will press on the Q&A button on the toolbar to expand the Q&A window. Type in a question, choose all panelists, and press the send button. We prefer to get the Q&A via that route rather than the chat. It's a little bit clearer. We will hold the questions for the end, and we will ask the panelists the questions that you might have. So just to walk you through that, there's a question button, click the Q&A button, type in a question, choose all panelists, and press the send button, and we will compile those questions. If you have any issues with the video or audio, I have the WebEx technical support phone number at the bottom, or you can send a Q&A, and we have a host from WebEx that's on the line to help with those issues. So today's webinar is using stem cells and high-content screening. I wanted to introduce you a little bit to high-content screening, what Medical Devices has to offer. High-content screening enables a number of applications, hundreds of applications, at a variety of optical resolutions, from receptor endocytosis assays, where you're trying to quantify the little dots in cells, angiogenesis assays, counting large objects like spheroids, high-resolution assays like mitochondrial formations for genotoxicity assays, and neurite outgrowths. Most of the talk is going to be on doing neurite analysis, but high-content screening allows you to do a variety of mitochondrial assays across in a screening setting. What's the typical high-content screening workflow? You have to acquire your images. You do some type of image analysis to convert those images and segment those objects into numbers and intensity measurements and shapes. You have to do some type of data analysis, and you have to have some type of storage management to handle all the images and measurements that are coming off the system. Medical Devices offers a complete solution for this workflow. With on the top left, you'll see the ImageXpress microsystem, which much of the work that's going to be presented on in the subsequent talk was collected on. We also have which is a wide field system. We also have the ImageXpress Ultra system, which is a confocal high-content screening system. On the right top, you have the image analysis tools that help run those systems and convert those images into numbers with ImageXpress and ImageXpress PowerCore software. And we have a QExpress software to help find those hits and graph those data to help with hit selection. Everything is tied together with a data management solution called Entity Store that gives you that complete high-content screening workflow. Mostly you're going to hear about data collected with the ImageXpress Micro, analyzed with ImageXpress, and some graphing of that data. Okay? So at this point, I would like to turn over the WebEx to Susan, who's going to talk about about the stem cell model that EDI has developed.
Thank you, Grischa. As Grischa has just said, my name is Susan DeLaura, and I'm from Cellular Dynamics. And my part of the talk is going to be discussing basically the human iPS cell-derived neurons and a way that, to show that they are physically relevant models to be used for the drug discovery. So just as a little bit of an introduction here, the nervous system is very difficult to model and very difficult to assess because of the complexity of it, especially when you're looking at also things that seem as simple as the neurotoxicity. It's an issue with the drug development as the fact that it has a notoriously high failure rate of only about 3 to 5% of the CNS candidates ever make it. Yes?
I'm sorry. We are not seeing your desktop yet.
Oh, I'm sorry about that. One moment here. There we go. How are you?
Yes, ma'am.
Okay, sorry about that. Okay. So, as I said, only about 3 to 5% of the CNS drugs are ever marketed, or that ever make it to the market even. Part of the issue is also the need for having a better model for being able to predict the physiology and the neurotoxicity earlier, and this will help improve not only the time to develop the new drug, but also improve the cost of it as well. Stem cells are a way that you can get that biologically relevant model, and it can be used for pretty much all steps in the drug discovery process, from the disease modeling, discovery, toxicity testing, etc. And when you combine both the stem cell technology along with high content analysis, you really end up with a very powerful tool or powerful way of being able to look at the compound screenings in a more physiological relevant fashion in both biochemical and cell-based assays. So for my part of the talk now, I'm going to just give you a brief overview of the company and our manufacturing procedure of the iCell neurons, which are the iPSC-derived neurons. And then I'm going to go over some of the characterization of our iCell neurons, and then I will pass it on over to Jayne Hesley for the high-content screening and assay development portion. So very briefly, Cellular Dynamics is, for anyone that doesn't know, it is a company that is, we make human iPS cells and iPS cell-derived cell types. And we are located here in Madison, Wisconsin. We have kind of four main core competencies that we focus on. One is the creation and culture of the human iPS cells from both normal and disease phenotypes, as well as doing genetic engineering of these iPS cells. So being able to introduce lineage and pathway-specific markers that will allow for more utility of those particular cells. We also have development of new differentiation protocols so that you're able to have these terminally differentiated cell types of interest. We can do it from all three germ layers, and we're able to scale up and manufacture these cell types to extremely high levels, and I'll speak to that a bit more in an upcoming slide. The products that we currently have are the iCell cardiomyocytes. That was our first cell type that we introduced. We also have iCell endothelial cells and our iCell neurons, and soon to come are the iCell hepatocytes. As I said, these are all human iPS-derived cell types. We also offer custom services for custom iPS cell reprogramming, and we can use a variety of different methodologies for doing that reprogramming. and as I said, then also the genetic engineering and differentiation. For the manufacturing process, part of the thing that we recognized that you had to do in order to get people, in order to make the cells work for the purpose that we wanted them to work for, we knew that you needed to have very high quality cells, very high pure cells, and also large quantities. And this is something that we focus very, very carefully at with the quality, quantity, and purity. The purity comes really into play when you're looking at cells that are terminally differentiated, because if you have any sort of a proliferating contamination there, it will quickly overgrow your culture, and then you're not actually seeing the results in your test from your cell type, but from that contaminant cell type. So we're very careful about being able to get high quality and high pure populations, and we do this with some of our cell types by using genetic engineering, proprietary method of doing genetic engineering to introduce purification methodology. This is a process that's not only able to be scaled up, but it can also be scaled out. And what I mean by scaled out is you can make the iPSCs from thousands of different individuals. So you can have a specific disease type that you're looking at or interested in, and you can make the iPSCs from that, and then further differentiate them if needed. And so therefore you're able to model many different types of diseases that are of interest. Our current product format is that we have, as I said, the human iPS cell-derived cell types that are cryopreserved and then just ready to use. With all of our products, we do offer at least a medium or a medium supplement that is there to ensure that the cells are able to perform to their highest capability. In the case of the neurons, we provide it with a medium and a medium supplement, allowing you to be able to grow the cells in culture for a maximum of, or a minimum, I should say, of about 3 to 4 weeks. So to focus a little bit more now on neuron characterization, as I said, we knew that there were certain things that we had to meet to really convince ourselves as well as others that the cells that we're producing are truly neurons. So some of the things that we looked at and had assessed were the morphology and making sure that it looks like a neuron. Some of the different molecular markers. This is a short list of some of the molecular markers that we looked at to make sure that our cells were expressing them. From your more general neuron marker of beta-3 tubulin down to a little bit more specific markers like your CGAT and your V-glut and some of the other post and presynaptic markers. We also were looking at functional characteristics, so wanting to look at the receptor and channel activation and the electrophysiology, and as Grischa had talked about what some of the high-content imaging assays are, the neurite outgrowth and sprouting. So for the morphology, if you look at the pictures here, you will see that the cells do indeed look like neurons. They grow to very nice neurites in a very quick fashion, and many of them they're at least bipolar, but many of them are multipolar, have multipolar neurites. The day one, 24 hours, you can see that we have a good amount of the neurites that have already grown after the cells have been thawed, for 24 hours. You actually start seeing that the growth of the neurons or the neurites at around about 4 hours after it's thawed, that you can already start seeing them. And as the days go on, you see that complex, the neurite complex becoming more and more dense. When you look at the purity of our cells, one of the things that we assess purity by is looking for cells that are making sure that they are beta-3 tubulin positive and nestin negative. The nestin negative is important because We are not wanting the cells to be containing a population of the neural stem cells or progenitor cells. So we're looking for these terminally differentiated mature neurons. Frequently, our cells are between somewhere around 96 to about 98% beta-3 tubulin positive, nestin negative. Cells are at a very high purity, and this is determined by flow cytometry. We also then looked at some of the gene expression of the protein, or of the neurons, and what we found with from the qPCR, just using a TACMAN array, is we found that the cells seem to be a population that is indicative of the forebrain identity. As you can see here, the regional gene expression, you have your high expression levels of the FOXG1, the DACH1, etc. So you have some of these forebrain identities. As far as the subtypes of the genes, you have your GABAergic and your glutamatergic are the predominant expression genes that are expressed. It does look as though there is a fair amount of the tyrosine hydroxylase for your dopaminergic neurons. However, it doesn't really translate to this amount when you're looking at it in the actual culture by ICC. It ends up being a very small scattering of the dopaminergic. And then as far as some of the receptor gene expressions, we looked at a variety of them. These are some of the main ones that people are frequently asking, and we have good levels of expression of various receptors. I will talk a little bit more about that in a couple slides coming up here too. So what this all came down to, what it kind of boils down to, is that the population of cells that our iCell neurons consist of is primarily the GABAergic and glutamatergic, around a 50-50 mix or 40-60% mix, meaning 40% glutamatergic, 60% GABAergic. And like I said, there's a very, very small percentage, less than 1%, that are the dopaminergic neurons. You can see from the ICC staining here, you see real nice VGAT and V-glue staining along the processes, as well as then this is just a picture showing some of the other, another type of a presynaptic marker, which is the synaptophysin, and in conjunction with the VGAT and the V-glue, and what you're seeing is some nice co-localization there. So you know that machinery's there as far as the synapses go. And then this picture is just showing you some GABAergic along with the MAP2 staining of the neuron. As I said, for some additional information looking at gene expression, this work was done by Actilian, and they were looking at using our neurons, and they wanted to compare it with the primary human fetal neurons, using with and without mitotic inhibitor, as well as comparing ourselves with the primary rat and a primary mouse at day 16 and day 18, respectively. So they wanted to see if our cells also, as the expression level of the receptors change over time. So looking at day 7, 14, and 21. And they were doing a calcium response using Slipper to determine the level of expression. What they found here was, this is just listing some of the different receptors that they were looking at and stimulating with the various ligands. And the primary message that they found from this was that the iCell neurons really did not change a lot over time from the day 7 to day 21. And they seem to correspond more to, or more similar to the mouse neuron cultures, but that the thought there was probably because the mouse neurons had a lower number of astrocytes there. Our cells do not contain astrocytes, and the astrocytes may have contributed to the signals of, for example, the acetylcholine and the serotonin receptor ligands. So as I said, these are a very pure culture, and the expression is going to be slightly different than some of the primaries from the standpoint that it does not have the glia cells present. They also used Agilent's whole genome array to look at our cells, and they were looking at the 397 gene set from the neurological system process to compare ourselves again with the primary human neuron. And basically one of the things that they found was that our cells were highly expressing the ion channels, glutamate receptors, and synaptic genes. They express lots of the GTCRs and very low glial genes, as we had said, that they are not present in our culture. We also then looked at the electrical functions of our cells and to make sure that they are functionally active. And this was using various methodologies. We used single-cell patch clamps as well as MEA, and it's the multi-electrode array. And as you can see here with the action potentials from the single-cell patch clamp, we're able to get very nice evoked action potentials with the nice overshoot and the rapid repolarization, as well as nice spontaneous action potentials. We're also able to look at or get responses to the ion channels and block them. So there's sodium, potassium, and calcium, and we can block them with the appropriate blockers. And then down here it's just showing an example of different types of profiles that we get on the MEA with our neurons. These are from 3 different electrodes as an example. And this is just a kind of a heat map in a sense of the neurons actually firing while on the MEA. We also did look at some of the postsynaptic currents as well by single-cell patch clamp analysis, and we are able to elicit excitatory and inhibitory responses using the appropriate agonist and block these responses as well with the antagonist. So the cells are electrically active and functioning as you would expect them to function. Another thing that we knew the cells could be very useful for is, for disease modeling. And in order to do that, sometimes you may need to do transient transfections or transduction of specific target genes. So we've looked into the capability of the iCell neurons, whether or not they're able to be transfected or transduced, as neurons are typically known to be very difficult to transduce or transfect. We are able to get transfection of the cells, the transient transfection. In this case here, it's just showing using GFT that's being constitutively expressed after the transient transfection. And for this, we were using the lipofectamine 2000, and typically getting right around that 30-ish% to 40% transfection efficiency. We're also able to do transduction of the target gene. We can get expression of the RFP that was in this particular construct here, up around that 65-75% efficiency. And as well as it is able to be used for backman transduction as another option. Another way to assess the health of your cells and even toxicity would be to be looking at the metabolic profile of the neuron. And in this case here, we had used the Seahorse platform, the Seahorse SF96 platform, and it's just another methodology of looking at mitochondrial health and function. So you are able to look at the oxygen consumption rate and see how the various compounds may affect the overall consumption, oxygen consumption, as well as you can look at the ECAR, which is the extracellular acidification rate, and monitor the glycolytic pathway. The cells are also very useful in doing, another type of toxicity testing. This is showing it being used for potency testing of botulinum neurotoxin. There is a paper out that describes this whole process, and it was a paper that came out of Eric Johnson's lab at University of Wisconsin-Madison. And basically, one of the things that they found when using the cells for this particular potency testing assay is that the neurons, our iCell neurons, were more sensitive than their standard cell type that they were using, which were the rat spinal cord neurons, or rat spinal cord cells. So it made for the potency testing assay to be a more sensitive assay. To switch over then to high content screening and assay development, as I said, this is a way that a lot of makes for an easy way of assessing toxicity of compounds on our cells. And the high content screening allows for a lot of capabilities, as Grischa had shown. But the one image that I was going to show is this particular one, which is just showing, which Jayne will talk a bit more about, but it's showing basically how you're able to see your neurons very well by ICC, but then the high content imager is able to map all of the neurites to the particular cell body, and it allows you to assess the overall neurite outgrowth and number of branches. And in this case here, it was just using BDNF or another growth factor type to see how it may enhance and increase that neurite outgrowth. And you can see that it made a significant difference over a 2-week period. The number of neurites grew much more densely. And with that, I'm going to turn the presentation over to Jayne Hesley for her to dive more into other high-content image-based assays.
All right. I believe I'm sharing my desktop right now, and everyone can see it. Speak up if that's not the case. I'm going to talk about some toxicity assays that we've done at Molecular Devices using the iCells from Cellular Dynamics. As Susan already gave you a good background about the nervous system and neurons being a target organ for toxic effects of a lot of chemical compounds, and we know that there's a need for predictive cell-based models to test stuff such as the toxic side effects of drugs, or maybe rescue the neurons from toxic effects, or look at the effects of different growth factors. What we offer is the opportunity to scale up your assays into high-content screening. All of the assays I'm going to show you were done in microplates. These are 96 or 384 well plates with our ImageXpress microsystem, which enables you to do automated acquisition. You can do multiple sites per well, multiple Z-stacks per well, and also once you've collected your images, we have automated analysis, both image analysis and then data analysis with the numbers that come out of the images. I'll talk about a few different assays. I wouldn't say we've spent a long time on every single one. We've shown definite feasibility for overall live-dead assay, doing neurite outgrowth assays, which can obviously give you more information about cells such as neurons. You can get a lot of information about the neurites, their length, their branching. A well-accepted assay for testing the integrity of your mitochondria. There are many drugs that affect the mitochondrial membrane potential. And then I'll show you some live cell assays that we did on our instrument as well. First of all, the live-dead determination. We did it 2 different ways. One is using a guy called Red Dead from Invitrogen, and it is showing you in the image on the right that your nuclei are stained with blue if they're alive, and if they're dead, they're stained with red. It's a very simple assay. We did this at a low magnification. 4x objective was used. You can count many neurons in one field of view to get statistically relevant numbers. The module for identifying these cells, one of our software modules, ImageXpress, is shown on the left, which allows you to just set up a few simple parameters. You choose the dye wavelength that is for the live cells, and then you choose the dye wavelength that's for the dead cells, and you do a few measurements to see what size they're going to be or what brightness they'll be above background, and then the software will identify all of these for you and all of the wells automatically. In the end, you end up with a heat map. You can look at it in many different ways: percent live, total live, dead, ratio, things like that. Another way we did the live dead determination was using the live cell stain calcine AM. We did this on live cells. So we identified all the nuclei with the HERC stain. Then the calcine AM will fluoresce only in the cells that are alive. You can score the cells based on the presence of one or both of the stains. So basically, how many of the cells are red, how many are red and green. So nuclear stained and as well as live cell stained. This one we did with the 10x objective, and again, you get many, many cells in every field of view. This shows a dose response to a toxin, methylmercury, where with a very low dose, your neurons are very much largely alive, and then as you're progressing higher and higher doses, you have many more dead neurons. We used a cell scoring module for this one, where we identified the nuclei, and then which of the nuclei also had green live cell stain. So in this overlay, this mask, it shows the red nuclei are from the dead cells. They had no live cell stain associated with them. The green nuclei were the live cells. You can do this across the whole plate, and we tested several different compounds, doing a 10-point dilution series with replicates of each one. This is the beauty of automated imaging. And we tested several compounds that were known to have neurotoxic effects in vivo, and they did show a dose-dependent toxicity. We also tested some compounds without known neurotoxic effects. The data is not shown, but such as aspirin and ascorbic acid, and we saw no toxicity at those levels, the levels we tested. To get more information out of your assay besides just live dead, you can do a neurite outgrowth assay where you stain the neurons for the neurites, the outgrowth. And this image clearly shows you that in live cells, untreated cells, you see a lot of outgrowth from each neuron. In staurosporine-treated cells, at a high level, the neurite outgrowths are definitely affected. Now, there are many drugs that might not necessarily kill the cells, such as we saw in the live dead assay, but it would definitely affect the neurite outgrowth. And another thing about neurite outgrowth is that this module can run very well with cells that are either stained with a fluorescent stain or in unstained cells. You can do it with bright field imaging. Here is an example of a bright field image with the mask overlay that has identified the neurons and the outgrowth. So the red that you're seeing is the mask showing the identified cells. The module is shown on the right. Again, very simple to set up. You just choose your parameters, whether your cells are going to be illuminated with fluorescence or whether you're just going to use transmitted light. And then you set up some identifying measurements, such as the size of your cell bodies, the kind of outgrowths you expect to, you would like it to identify, if there's a minimum length you want it to find, and whether or not you have a nuclear stain. This works well without a nuclear stain, too. The parameters you get out are multiple. You can get the number of processes per cell. You can get the length, the branching, the overall number of neurites. You can do it per field of view, or you can do it per cell. As you can see in this table, on the left, cell assigned label number is each individual object that's measured in the field of view. Another thing we can show is that if you image your cells and you really want to follow the neurites, they're very long, and you want to measure the ones that would maybe go out of a field of view, you can capture multiple fields of view and stitch them together into a large field, into a large image. Then this large image, this one I'm showing you is 12 images taken and stitched from a 10x objective. Then you run the analysis module on it and you identify your cells. Now I'm going to show you the segmentation on a smaller area. Here are the cells in that small area, zoomed in, and the segmentation that we found. So it can identify your neurites very well. I'll talk a little bit. We also have an ImageXpress micro XL system. XL is the system we have that has a more than a 3 times larger field of view than the standard system that we've been selling. The XL has been introduced about the end of last year. And so you can already, without stitching, get 3 times more cells in one field of view, which enables a really significant number of cells to be tested. Another example of a standard curve using the neurite outgrowth, so similar to what we saw with just the live dead assay, measuring the branching or the total number of outgrowths or the percent of cells that have an outgrowth. We tested several different drugs, and we calculated different IC50s based on some of the different parameters we measured. They agreed with each other pretty closely. You can see in the table on the right, antimycin A, whether we looked at the total number of neurite outgrowths or the percent of cells with outgrowths. The IC50 we calculated was still right around 13, 14 micromolar. So another kind of assay we did was monitor the mitochondrial membrane potential. So this mitochondrial integrity is an early indicator of cell distress. We use the JC10 reagent, which is similar to JC1, if people are familiar with that, more soluble though. And it selectively accumulates in the mitochondria as orange J aggregates. So the color will be orange or something in the Texas red channel if it's maintained inside the healthy mitochondria. If the mitochondrial membrane potential is disrupted, the dye leaks out and it fluoresces green in the cytoplasm. So just remember, the healthy cells, healthy mitochondria, will have orange mitochondria showing, and the unhealthy cells will have few orange mitochondria showing, but green will appear in the cytoplasm. This can be run as a live cell assay. You could even run it as a time course, or it can be run as an endpoint assay. This dye is maintained for a period of time after the cells are fixed. This shows an example of the cellular dynamics neurons with, on the left-hand side, with healthy mitochondria and untreated control. And I hope you can see the puncta mitochondria in the neurites. And then on the right-hand side with a high dose of antimycin A, which is a compound that's known to inhibit mitochondrial respiration, that you have very few mitochondria being shown in the orange, and you have a lot of green unhealthy cells. The effects of this are visible within about 30 minutes. 30 minutes is the incubation time for the JC10. You can put it on, and the instrument has an option for environmental control, so you can maintain your cells at temperature and CO2 control, and you can take a time course if you'd like of this. But within 30 minutes, this is what the effects look like. These were taken with a 20x objective on the ImageXpress Micro. Now we can use our module called granularity for counting the mitochondria, and this is what we did, so we could quantify how many mitochondria were in the cells. And this graph shows two compounds that are known to interfere with mitochondrial membrane potential, antimycin A and valinomycin, and you can see a dose-response curve over time in the neurons. So I talked about doing the live cell assays. We did an experiment where we plated the cells directly from thawing and watched them grow overnight, basically. So these were started about 2 hours post-plating, probably, so they had time to sort of attach. And the control wells, as you can see, when they're at the end of about 14 hours after plating, so after 11 hours of live cell imaging, you can see a lot of neurons growing out of those. Cells that were treated with staurosporine, also, these are all on the same plate, and it definitely inhibited the neurite outgrowth. They were not happy cells. They were not healthy. So this was done in a 96-well plate where we imaged every 20 minutes, every well every 20 minutes. And so these are the videos that are created from the time-lapse images. So what we did, I guess I explained, we did it with transmitted light. It was time-lapse acquisition up to about 14 hours post-plating, and then we did automated analysis on this. So you can see that we identified the neurons and the neurite outgrowths in all the wells, but I'm showing you the ones that have the biggest difference with untreated and staurosporine treated wells. At the different time points, you can see how much they've grown at time 0, then 3 hours, then 9 hours later. And we plotted this on a graph showing that staurosporine significantly inhibited the mean outgrowth per cell. Again, we did this on the ImageXpress microsystem that we sell here at Molecular Devices. We acquired the images sometimes 4x, 10x, or with 20x objectives. The images were analyzed using our ImageXpress software. I've shown you data from the neurite outgrowth algorithm in ImageXpress, granularity, also the self-scoring, and the live dead modules from ImageXpress. If you do ImageXpress Micro XL, there's a much larger field of view, and you get a much greater sensitivity and dynamic range. Thus, I've demonstrated the assays, the live dead, the neural network integrity, mitochondrial integrity, and the cell or neuron outgrowth over time. The other thing is that those assays could be multiplexed. The analysis of those assays can be multiplexed in our ImageXpress software. And you could get, multi-parametric data output. And could be used for obviously testing compound effects on neuronal development, screening and validating drug candidates, and evaluating potential neurotoxic effects of different agents. I think using cells such as neurons that are relevant cells will help scientists to avoid false drugs going past an early stage. Anyway, I'd like to thank from Molecular Devices Oksana Serenko for doing a lot of this work on the ImageXpress Micro, Penny Tavermina, and then from Cellular Dynamics, we've had a really great collaboration with their scientists at Cellular Dynamics, Lucas and Monica. David and Jeff given us a lot of advice on using their cells. And with that, I will pass the ball back to Grischa so that he can wrap up and we can answer questions if anybody has any.
Thanks, Jayne and Susan. That was great. Actually, there's a few questions that come up about how data was acquired. That leads me to I'll give an introduction to our new product and talk about some of the capabilities that will maybe answer some of the questions about how these data were acquired. I hope that people are seeing my presentation. Okay, so I'm going to switch back to the host role and just give you a little reintroduction to our product. So most of these data have been collected with the ImageXpress microsystem, and they've been using our older standard model. We do have a new system that Jayne hinted at, the ImageXpress Micro XL, and also that product was launched with new software on the ImageXpress side. People asked what magnifications the data was generated with. Most of the data, I believe, that Jayne collected was either at 10 or 20x. The system is capable of being configured with a very wide range of objectives from 1 to 100x. Okay, but for these assays, most of them were in the 10 to 20x range. And at the top of the Q&A session, we'll have Jayne comment on any deviations there. Let me pass this away. I did want to mention a little bit about the XL. We have launched a newer product that increases the field of view that you can capture in one image. So on the left is a standard system with the green square being the field of view within a 3D 4-well plate. And on the right is what you would get with the XL system. You'd be able to, with a 4x objective, be able to capture a whole single 3D 4-well in one image. So for neurite analysis, that is quite advantageous because you can reduce the number of images you collect. is also important for the assay. Also, the ImageXpress XL system comes with a solid-state light source that gives you on-demand illumination and reduces some support requirements because you don't need to change a light bulb or replace the mechanical shutters. Those are the two big component changes, but what that allows you to do is significantly speed up your acquisition by about threefold, because oftentimes with a standard system on the left, you're getting about 120 cells per field, and if you want to capture most of a well, and this is for a cancer objective, you have to go up to 50 images. Whereas with an XL, if you try to capture the same area, you'd only have to capture 16 images. Now, these are extreme examples. The example that Jayne gave, her 12 images would drop to 4 images. And as a guide as how long this would take, it takes about 4 minutes to acquire the two-color assay in an iCell plate. Okay? And you maintain or increase your assay quality with the product, with the Z prime going from about 0.6 to about 0.7. Additionally, your system uses a scientific CMOS as a detector, and that improves the dynamic range of your images and helps with some assays. On the left, you have an image captured with the XL. On the right, you have the image captured with the standard. And if you do a line scan through that, you'll see that sometimes if you put it on a very big scale, it's hard to see those dim neurites with those high bright cell bodies. Sometimes they'll overexpose the cell bodies to see their neurites. But with the XL product, with the scientific CMOS with a larger dynamic range and 16-bit digitization, you can see those very dim neurites in the background of bright objects. So that's the cell body, and those are the outgrowths. So it's easy to resolve fine features in the presence of bright objects with the new XL product. Additionally, we improved the speed of ImageXpress natively, and also ImageXpress modules can run on a tool called ImageXpress PowerCore. Just to give you a hint that when you're trying to do high throughput imaging, when you're trying to collect these big images across multiple plates, you need your speed of analysis to keep up with your speed of acquisition. And we have a way to run each of those modules across multiple cores of a computer, and your analysis time drops significantly depending on the number of processes that you have to run the module. So your analysis can be done in minutes and keeping up with the acquisition. So just to introduce to some of the people unfamiliar, we have the ImageXpress Micro Scanning System, and we've recently launched the ImageXpress Micro XL, which allows you to run more screens, perform faster, maximum uptime, maintains high quality images, and lets you grow with your expanding applications. And if you want to learn more, you can go to highthroughputimaging.com. So please continue to submit questions. I will have compiled a list of a few ones that I will ask Jayne and Susan, and send those questions on to us. Just one of the key questions that have come up is, will the webinar be available later? For those who register, we will send an email out to them with a link to a recording of this webinar within a few days. And let me switch over to Q&A. Can I share? Sorry, I just need to go pull up the question out of Susan. One of the questions was, it was about, I think you touched on it, but maybe you can expand. There was a question about purity and whether you've looked for quantification of glial cells that come up in cultures, and specifically GFAP. Okay?
Yes, I've got a couple different questions jotted down here that I that I've taken off of the panel or off of the Q&A. One of them was, yes, wondering about if we stain specifically with our neurons for the GFAP, and wondering about if, like, the glial cells start to appear over time or develop over time. The answer to that is yes, we have done staining as well as flow for GFAP with our neurons. And we do not see any staining at nor see it by flow right out of cell or over time. We have cultured the cells, continued to culture the cells and then reanalyze it, I believe, at days 14 and 21, and we still don't see any presence of the glial cells there. Another question that I had was what type of serotype of the AAV was used. We specifically used in-house here ourselves the AAVS1. I know that some outside users, some people that are using our cells have also used a mix of 1 and 2, and it has been successful with that. Another question that I did see was how long can I expose the neurons to compounds? The neurons, really is going to be dependent on your assay type as well as exactly how toxic the compound is to the cell. The neurons, we've kept them in culture way past 28 days. We've looked at the functionality of them definitely out to 28 days and a little bit beyond that as well. However, we've also kept them in culture and they do stay viable out past, I think the longest that we kept them in culture and then finally just got rid of them was like around 9 months or so. So we hadn't done all the functionality or characterization of the cells at that point. But definitely, if the compound is not too toxic to the cells, and depending on your assay, you could, keep them in culture for, 4 or 5 weeks or so, and the functionality should still be there.
Thank you, Susan. A couple of questions for Jayne. I think you mentioned you did mostly 96 and 3D4 plates, is that correct? And did you treat the plates with anything, or what was the protocol used to get the cells down and get them imaged?
Yes, I followed the protocol exactly from Cellular Dynamics for coating the plates. And I can't exactly recall, but Susan, I'm sure knows.
Jayne, let me interrupt a minute. The plating, the coating that we typically use is a double coat of PLO, polylysine ornithine, and followed by an overcoat of laminin. Right.
This is what we did, and you can use fairly standard plates from the common vendors. I think we've used CD and Corning plates both. So 96, 384 well plates, readily available. The cells coat very easily down on the plate. And yes, certain types of coating can give you, I know some people have seen, I don't know, when you do higher magnification, some waviness, kind of, in the images, but we have no problem still analyzing those, at least.
Great. So there's a question about, so I think you've been doing can you talk about the number of colors that you used in the assays and what the number of colors used in the assays presented?
We used Herbst for nuclear stain. We've used several different of the Electrofluor secondary antibodies, 546, 647, 48. We've used calcium AM and the JC10 reagent. We've used Mitochondrial Red.
Right. So to answer the question online, so the system is capable of taking data from a number of resin dyes and as well as bright field. And at any one time, you can people have acquired up to 5 colors if you pick the right resin fluorophores. Usually people are doing things in the 2, 3, and maybe 4 color range. I'm just screening
That's exactly true, yeah.
Yeah. I'm just sorry, folks, I'm just screening through to make sure that I'm looking at the other questions. There's a question about how many neurons can get plated in a well. Maybe you can talk about how many get put down, but typically we are imaging with the XL, with the 10x objective, you're seeing about 500 cells.
Or more.
Or more, yes.
It's going to depend, yeah. It's going to depend on your plating density and everything, of course.
There are some questions about cost of the system. My email address is grischa.chandy@moldesk.com. It should have been on the invite. Please send that to me by email, and I'll forward that information onto the sales, onto sales. So if anybody's interested in pricing of either the instrument or the CDI cells, I think the CDI cells, you can also contact me, and I'll forward that on to Susan. I think, unless there are more questions, we are about to wrap up. I'm just doing one more scan of there's a question here, do we ever test accuracy of the neuron outgrowth with the various staining, 2-to-1 staining or bright field, or I guess even calcium AM. Jayne, would you comment on that?
What was it, accuracy?
Yeah.
Like compared to each other?
Compared to each other.
You know, I think you probably get very similar results. I've seen the calcium AM stains the neurite outgrowth very well. And the 2-to-1 certainly does. And we have bought a directly labeled 2-to-1. The beta tubulin, Alexaflor 488, that works very well. It gives you much less wash death when you don't have to add the secondary antibody.
Great. So, is there advantages of using glass bottom plates? Should I take that one?
Yes, if you want to from the instrument.
So I would say for the
point of view might be different.
Yeah, so I think it depends on the glass bottom plate, depends on your assay and the resolution you want to image. We are one of the ACS systems on the market with the most flexible systems, meaning we can take nearly any type of clear bottom plate. Glass bottom allows you to use higher quality, higher numerical aperture objectives, and that's useful for sensitivity and resolution, especially at higher magnification. At the lower mags, there might not be as much advantage of using the glass bottom plate.
And also, I want to add the caveat to that is going to be you'll need to do some development with your cells, because many cells do not attach and grow as well in the glass bottom plate.
Great. So I think we're a little bit... Oh, go ahead, Jayne, sorry, go ahead.
No, I was going to mention also, if there are people out there, we've done these neuron assays in lower density well plates from 96 also. We've done them in 24 well plates as well. I'm not sure if you mentioned them, so I can definitely read those. They don't necessarily have to be SBS standard plates.
Yeah, I did not mention the lower density plates. Thank you. Okay, it looks like we're just over after 9, and I'm not seeing any other questions popping up. If there are any technical questions, I see there's some from users of our product. Please just drop me an email and I'll forward it on to my people. If there's questions about pricing, the same. Susan, do you want to leave with any last words?
Yeah, I was just going to say, if anybody had a question about the iCell neurons, you can directly email me at DeLaura, it's DeLaura @cellulardynamics.com.
Good. Thank you very much, everybody. Thanks for attending. Please come to our next webinar. We'll have one in a couple of weeks on screening with RNAi, and that should be very exciting as well. Thank you. Bye-bye.